So it's a late afternoon in September of 1848, and there's this railroad foreman named Phineas Gage. He's out there setting explosives into a rock
Right.
face.
A very routine job for him, usually.
Yeah, exactly. And he's using this custom-made iron tamping rod. It's like 3 feet long, over an inch thick, and weighs 13 pounds. And he's using it to pack explosive powder down into this deep hole.
Which is incredibly dangerous work, even on a good day.
Seriously. But he gets distracted. He turns his head for just a second, and the iron rod strikes the rock, and it creates a spark.
Oh, wow.
Yeah. The powder detonates prematurely, and this massive, heavy iron rod shoots out of the hole like a rocket. It enters right under his left cheekbone, passes behind his left eye, and literally explodes straight out through the top of his skull.
It's just horrifying to even picture.
It really is, and it lands, coated in blood and brain matter like 80 feet away.
And I mean, what makes this specific case such a cornerstone of biological science isn't just the gruesome nature of the accident.
Right.
It's the fact that Gage didn't actually die. I mean, within minutes, he was sitting up in a cart, entirely conscious, just chatting with his crew on the way to the town doctor.
Which, completely shattered everything science thought it knew about the human brain at the time. And that is exactly where we're starting today's deep dive.
It's the perfect entry point.
It really is. So whether you are out there currently cramming for some brutal anatomy exam or trying to catch up on human biology, or, you're just deeply curious about the biological engine running your own mind, our mission today is to shortcut your way to understanding how it all works.
And we have an incredible stack of sources to pull from today to do that.
We do. We've got this comprehensive instructor's manual that's based on a monumental psychology textbook chapter covering the brain and nervous system, plus some wild historical clinical case studies like the Phineas Gage one.
Yeah, and we've also got a really fascinating batch of cutting-edge educational research from early 2026.
Right. So, to kind of frame our goal for this deep dive, there is this brilliant quote in the textbook from the pioneering neuroscientist Santiago Ramon y Cajal.
Oh, yeah, I love this quote.
Yeah.
He observed, the brain is a world consisting of a number of unexplored continents and great stretches of unknown territory.
Unexplored continents. I just love that phrasing. Our mission is to basically map that territory for you today. And Phineas Gage was essentially the first brutal, accidental attempt at that kind of cartography.
He really was.
So let's look at why Gage actually survived, but also, what he lost. Because the rod passed through the very front of his head, it entirely missed the brainstem. That's that primitive structure deep down at the base of the skull.
Exactly. And the brainstem is fundamentally the biological autopilot. It manages all those vital functions you never really have to consciously direct.
Like breathing and stuff.
Yeah, respiration, heart rate, your basic arousal levels.
Yeah.
So because the rod spared that really deep structure, Gage's body just kept functioning. However, the rod completely obliterated specific cortical regions in his frontal lobes.
And while his heart kept beating, the man himself changed completely. I mean, the sources note he went from being this highly capable, mild-mannered, really responsible leader to someone who was deeply impulsive, profane, and just completely unable to regulate his emotions.
He couldn't stick to a plan at all.
No, not at all. His friends famously said he was, quote, no longer Gage.
Which is so tragic, but if we connect this to the bigger picture, this accident definitively disproved the old theory that the brain was just a uniform mass.
Oh, or they thought any part could just do any job.
Right, exactly.
Yeah.
Gage's survival demonstrated the localization of function. The primitive brainstem keeps the biological machine running, but those distinct, highly specialized regions in the outer cortex, they manage the complex, uniquely human behaviors, like personality and emotional regulation.
Okay, let's unpack this, because this macro view of the brain actually maps directly onto the larger architecture of our entire nervous system, right?
It does, yeah.
Our sources outline two major divisions. You have the central nervous system, or the CNS, which is the brain and the spinal cord. That is basically the central command center.
Right. And then reaching out from that command center, you have the peripheral nervous system, or the PNS.
The branching network.
Exactly. It's this vast communication network that connects your brain and spinal cord to literally every organ, muscle, and sensory receptor in your entire body.
And within that peripheral system, we have two subdepartments, I guess you could call them. There's the somatic division, which handles voluntary movement.
Like you deciding to reach out and pick up a coffee cup.
Exactly. And it transmits sensory data back to the brain. But then there's the autonomic nervous system.
Yeah, and the autonomic system operates entirely beneath our conscious radar. It manages the involuntary visceral functions necessary for survival.
Like digesting food or dilating pupils.
Right, keeping the internal organs functioning.
Mm.
And this system is divided into two highly reactive opposing branches.
So the autonomic system is kind of like a car, right? The sympathetic branch is the gas pedal for emergencies, triggering that intense fight or flight crisis response.
Yes, exactly.
And then the parasympathetic branch is the brakes, calming the body down for resting and digesting.
That's a great analogy.
But looking at the 2026 current events in our sources, specifically regarding academic stress, are we all just driving with the gas pedal slammed to the floor?
Honestly, the 2026 research indicates that physiologically, yes, we often are.
Wow.
The academic publications in our stack, they monitored diverse undergraduate cohorts and found that the relentless anxiety of modern education, constant exams, social pressures, financial worries, it triggers prolonged, chronic sympathetic activation.
So the body literally thinks there is a physical threat constantly present. Like a bear is chasing you, but it's just a midterm?
Precisely. And the biological mechanism here is basically a zero-sum game. When the sympathetic gas pedal is pinned down, it floods the body with cortisol and adrenaline, and that actively suppresses the parasympathetic brakes.
That just overrides the rest system.
Yeah, and the research specifically highlighted non-traditional learners too.
Yeah.
Students balancing full-time employment with heavy course loads.
Because they never get a break.
Right. Because they rarely experience true downtime, their parasympathetic system just doesn't have the opportunity to engage its restorative functions. And that leads to severe physiological and cognitive strain.
So all of this macro-level stress just completely overloads the systemic architecture.
It really does.
But our sources point out that to really understand how a thought or even a stress response travels through that system, we have to zoom all the way in. Like, we need to leave the whole brain behind and look at the microscopic level of individual cells.
Oh, absolutely. And the discovery of how these cells actually talk to each other is just as dramatic as the Phineas Gage story, though significantly less bloody, thankfully.
Way less bloody. It happened on Easter weekend in 1921. There was this pharmacologist named Otto Loewe, and he wakes up in the middle of the night with a fully formed idea for an experiment. He scribbles it down, goes back to sleep, and wakes up the next morning completely unable to read his own handwriting.
Which is just the most relatable thing in the history of science.
Right. But luckily, the next night, the idea comes back to him. So he immediately rushes to his laboratory to conduct an experiment on frog hearts that literally upended our entire understanding of biology.
Because prior to Loewe's midnight experiment, the dominant consensus was that neurons, which are the fundamental cellular building blocks of the nervous system, communicated purely through electrical sparks.
They assumed the nervous system was essentially just one continuous copper wire.
Exactly. But Loewe proved it wasn't just electricity. He isolated a frog heart with its vagus nerve still attached, placed it in a saline bath, and stimulated the nerve electrically.
And as expected, the heart rate slowed down.
Right. But here's the genius part. He took the saline fluid from that first heart and applied it to a second, completely unstimulated frog heart.
And the second heart slowed down as well.
Yes. By transferring the fluid, Loewe proved that the electrical stimulation in the first heart had actually caused the nerve to release a physical chemical into the saline.
So cellular communication wasn't just a continuous electrical spark. It relied on chemical messengers.
Exactly.
So mechanically, how does a thought or an impulse actually cross from one cell to another today? Like, how does that work?
Well, it is a dual process. It begins electrically and concludes chemically. Inside a single neuron, the communication is an electrical impulse, which we call an action potential.
Okay.
And this electrical charge is generated by a rapid exchange of sodium and potassium ions across the cell membrane.
And that electrical charge travels down the neuron's long cable-like structure, right, the axon, until it reaches the very end.
Yes, it reaches the terminal buttons at the end. But at this point, the electrical signal hits a physical chasm. Neurons do not actually touch each other.
They don't touch at all.
No. There's a microscopic gap between them, known as the synapse, or the synaptic cleft.
And the electrical spark cannot jump that physical gap.
It cannot. So the mechanism must change. When the action potential reaches the terminal buttons, it essentially forces these tiny saps to burst open.
Spelling those chemical messengers.
Exactly, spilling neurotransmitters into that synaptic gap. These chemicals float across the space and lock into perfectly shaped receptors on the receiving branches. the dendrites of the next neuron.
Kind of like a key fitting into a specific lock.
That's exactly how it works. Once those chemical keys turn the locks, it triggers a brand new electrical action potential in the receiving cell, and the message just continues down the line.
Okay, here's where it gets really interesting. Wait, so every complex human thought, every memory you study for an exam, every single emotion you feel is just chemicals floating across a microscopic gap.
Pretty much, yeah.
That sounds incredibly fragile. Can our daily habits actually disrupt this?
They absolutely can, and the 2026 research articles in our stack focus heavily on this fragility. Specifically, they looked at how screen-induced blue light alters these chemical pathways in college students.
Which is super relevant because everyone listening is probably staring at a phone or a monitor right now. So how does the light actually affect the synapse?
Well, the visual cortex registers that specific spectrum of blue light and sends a signal deep into the brain that it's still daytime,
Even at midnight.
even at midnight. And this suppresses the synthesis and release of melatonin, which is a critical neurotransmitter for regulating sleep cycles. So late-night studying on digital devices essentially confuses the chemical messaging at the synapse.
So you aren't just tired the next day. Your brain's chemical signaling is actively misfiring.
Exactly. And the downstream effects are severe. When melatonin pathways are disrupted, the brain cannot achieve the deep sleep phases that are required for memory consolidation.
Right, locking in what you learned.
Yeah. Building a memory requires neurons to strengthen those synaptic connections. It's a process called long-term potentiation. So, disrupting the neurotransmitter balance with late-night blue light physically prevents the brain from reinforcing the chemical bridges needed to master complex material.
So if learning and memory are entirely dependent on these delicate chemical bridges, what happens when the deep brain structures that organize them are damaged?
That's a great question.
Let's move from the cellular level to the subcortical level, to the parts of the brain that govern our most ancient evolutionary drives.
Yeah, and the textbook introduces a really fascinating clinical case study from the late 20th century to illustrate this. Researchers extensively documented a woman who was identified only as patient SM.
Right, she suffered from a rare genetic disorder, right? One that calcified and destroyed a very specific almond-shaped structure deep inside her brain called the amygdala.
Yes, and because of this highly localized destruction, patient SM experienced life entirely devoid of fear.
Which, I mean, sounds like a superpower at first.
It does, right?
But researchers took her to haunted houses, they showed her horror films, they even exposed her to live snakes and spiders. And she experienced curiosity, maybe mild excitement, but absolutely zero biological fear.
And this case demonstrated that the amygdala is fundamentally the neurological threat detection center. Without it, SM lacked the basic survival instincts that keep humans from, stepping into oncoming traffic or approaching dangerous predators.
Right. As the neuroscientist Antonio Damasio points out in the text, emotion isn't just a psychological luxury, it is a complex biological agent required for an organism's survival.
Exactly. And the amygdala doesn't operate alone either. It is a central hub within a much larger network called the limbic system.
Right, the limbic system.
It's structurally fascinating. Geographically, it forms a border like a ring between the primitive autopilot brainstem we discussed earlier and the advanced thinking cerebral cortex on the outside.
So it's the anatomical bridge between basic physiological arousal and complex psychological thought.
Precisely. It manages a lot of heavy lifting. Next to the amygdala is the hippocampus, which is crucial for converting short-term experiences into long-term memories. It also handles spatial navigation.
And then just below the thalamus is the hypothalamus, and that structure maintains homeostasis, right? Like the body's internal balance.
Yes, it constantly monitors your blood chemistry to regulate primary survival drives: hunger, thirst, body temperature.
It is basically the ultimate survival control room. But going back to patient SM for a second, if she survives without fear, does the amygdala just cause us unnecessary stress today?
How do you mean?
Like, we aren't running from predators in the woods anymore. So how do we calm the amygdala down when we are just overwhelmed by modern life?
Well, the challenge is that the amygdala is an ancient piece of hardware running in a modern environment. It often cannot distinguish between the existential threat of a charging predator and the social threat of a final exam.
It just panics either way.
Right. It just detects a stressor and floods the system with fear signals. But the 2026 educational psychology journals in our sources actually offer a biological counterbalance to this.
Okay, so how do we signal to the amygdala that we are actually safe?
Through structured social connection. Yeah, The research looked at campus community initiatives and found that peer mentorship groups significantly soothe limbic system hyperreactivity.
So just participating in a study group or a mentorship program physically alters the fear response in the brain.
Yes, it does. For both traditional and older, non-traditional students, intentional social integration signals safety to the limbic system. Evolutionarily, being part of a group meant survival.
Oh, that makes so much sense.
So when the brain feels socially secure, the amygdala actually downregulates its threat signaling. It reduces the production of stress hormones and fosters the emotional stability required to navigate a demanding academic environment.
Wow. Okay, so we've explored how the inner deep brain regulates internal emotions and threat detection. But how does the outer brain orchestrate outward physical action? We need to move up from the limbic system to that wrinkled outer layer of the brain, the cerebral cortex.
Right, and to understand the functional mapping of the cortex, we rely heavily on the surgical breakthroughs of the 1930s, specifically the work of this neurosurgeon named Wilder Penfield.
Penfield's work sounds like absolute science fiction to me. He was performing conscious neurosurgery to treat severe epilepsy, so he would remove a piece of the skull, exposing the living brain of an awake patient.
Which sounds terrifying, but because brain tissue itself actually has no pain receptors, only the scalp requires local anesthesia. So the patient is fully awake and conversing with him.
That is just wild.
Right. And Penfield would gently touch different areas of the exposed cortex with a small electrical probe just to see what happened.
And the results were crazy. He would touch one spot, and the patient would report smelling burnt toast. He'd touch another, and they would vividly hear a specific song from their childhood.
Yeah.
But when he stimulated a very specific vertical strip at the back of the frontal lobe, the patient's fingers would twitch or their lips would move completely involuntarily.
Penfield actually once described the brain as the organ of destiny, holding in its lines of force all the potentiality of existence.
That's beautiful.
It is. What he was meticulously charting with that electrical probe was the primary motor cortex. It's located along the precentral gyrus, and this strip basically manages all of our voluntary bodily movements.
But the way this strip is organized is what makes it so fascinating to me. It features a somatotopic map, often referred to as the motor homunculus.
Right. It is essentially a physical map of your body, but draped over the surface of your brain. Specific regions of this cortical strip correspond directly to specific body parts.
So what does this all mean? The textbook explains that the amount of space a body part gets on this map has absolutely nothing to do with the physical size of that body part.
Where it's not proportional to size.
No, it is based entirely on the precision of movement required. So this biological strip is like prime real estate, and the most precise skills get the biggest mansions.
That is an excellent way to conceptualize it. Think about it. If you shrug your back right now, you are moving a massive amount of physical muscle and bone. But your back requires very little complex control, so it occupies only a tiny sliver of real estate on the motor cortex.
Okay, yeah.
However, if you wiggle your thumb, or if you speak a sentence, you are executing incredibly intricate motor commands. Consequently, the hands, fingers, lips, and tongue receive disproportionately massive representation on the cortex.
It's basically a biological reflection of human capability, but is this real estate map set in stone from birth, or can we renovate? Like, can you build a bigger mansion on the cortex if you need to?
You actually can. The map is highly plastic, meaning it is structurally adaptable. The 2026 news reports in our sources document how vocational training rapidly remodels this cortical real estate.
Oh, through neuroplasticity.
Yes, exactly. The reports looked at first-year undergraduates engaged in intensive typing courses and hands-on laboratory workshops. By repeatedly executing fine motor skills, these students actually expanded the structural representation of their hands and fingers within the motor cortex.
Wow.
Yeah, the practical training physically alters the brain's architecture to accommodate new, career-driven tactile mastery.
That adaptability is just incredible. Now, Penfield had to literally open a person's skull to map that real estate back in the 1930s. But today, we don't need a scalpel to watch human thoughts and movements light up.
No, thankfully we don't. The evolution of neuroimaging technology in the late 20th century fundamentally changed neuroscience. It allowed us to observe the living brain in real time, safely and non-invasively.
But how exactly do we see through the bone of the skull without opening it?
Well, different technologies measure different biological signals. Electroencephalography, or EE, relies on electricity, When millions of neurons fire action potential simultaneously, they generate an electrical field strong enough to leak right through the skull.
Okay.
So by placing sensitive electrodes on the scalp, an EEG tracks these electrical waves millisecond by millisecond.
But an EEG just shows wavy lines on a screen, right? It doesn't give you a picture of the deep structures like the amygdala we were talking about.
Correct. For structural clarity, we use MRI magnetic resonance imaging. Your brain is mostly water, which means it is full of hydrogen protons.
Right.
An MRI uses a massive magnetic field to force all those tiny protons to align in one direction. Then it pulses a radio wave to knock them out of alignment. When the protons snap back into place, they emit signals that a computer translates into incredibly detailed, high-resolution static images of the brain's anatomy.
So that gives you a structural map. But what if you want to see the brain actually working? Like, where is the energy going when I solve a math problem?
That requires tracking metabolism. PE scans do this by injecting a harmless radioactive glucose tracer into the bloodstream.
Radioactive sugar.
Basically, yeah, active neurons are hungry. They consume more glucose. So the PE scan detects where that radioactive sugar is being metabolized, showing us the hotspots of neural activity.
Okay, and then there's functional MRI, or fMRI, which seems to be the gold standard in the textbook. How does that differ from a regular MRI?
Well, it relies on the magnetic properties of blood. Hemoglobin carries oxygen, and oxygenated blood responds to magnetic fields differently than deoxygenated blood.
Oh.
When a specific region of your brain is working hard, it consumes local oxygen. The cardiovascular system immediately flushes that area with fresh, oxygen-rich blood to restock the supply. And fMRI tracks that subtle magnetic shift in blood flow dynamically.
Oh, so you aren't actually measuring the thought itself. You are tracking the blood flow that feeds the thought.
That is a perfect summation of the mechanism. You map the demand for resources.
But, I mean, these all sound like massive multi-million-dollar hospital machines. How is this advanced technology actually being applied to help someone like you, the listener, who is just sitting in a classroom or at a desk trying to learn something new?
Well, the 2026 science report highlights a huge shift from the hospital to the classroom. Universities are now actually deploying highly portable, lightweight EEG headbands during actual lectures.
Wait, so students are wearing brain monitors while the professor teaches?
They are. The EEG headbands track aggregate cognitive engagement across the entire room. If the professor's explaining a complex theory and the EEG data shows engagement sharply dropping, the professor receives real-time feedback.
That is wild.
Yeah. They don't have to wait for a failed midterm to know the students are lost. They can immediately adapt their instructional approach, provide a new analogy, or just slow down the pace to re-engage the struggling learners.
The technology is actively adapting the environment to fit the biology.
That's amazing.
Yeah. We have covered an immense amount of ground today. We started with an explosive railroad accident in 1848 that revealed the localized architecture of our cortex.
We did.
We zoomed in on Otto Loewy's midnight revelation about chemical neurotransmitters bridging the synaptic gap. We explored how patient SM proved the amygdala's critical role in survival. We mapped the precision-based real estate of the motor cortex, and finally looked at how magnetic fields and portable EEG bands are decoding the mind in real time.
The terrain is vast, but as we said at the beginning, mapping this unknown territory is an ongoing dynamic process.
And understanding these biological mechanisms is the ultimate shortcut to navigating your own daily life and whatever pressures you face. Knowing why putting your phone away late at night physically protects the chemical pathways of your memory, or knowing how joining a mentorship group physically signals safety to your limbic system, that is real power. You are not just a passenger trapped inside your biology, you can actively work with it.
Which perfectly circles back to Santiago Ramon y Cajal's underlying philosophy, actually.
Yes, I want to leave you with one final thought based on Cajal's belief. He wrote, "Every individual can, if they so desire, be the sculptor of their own brain." So here's something for you to mull over as you go about the rest of your day.
Think about it.
If intensive typing practice can physically expand the real estate in your motor cortex, and late-night blue light can fundamentally alter the chemistry in your synapses, what intentional daily habit will you practice today to physically sculpt the unexplored continents of your own brain tomorrow?
The great question.
Keep exploring, and we will catch you on the next deep dive into the sources.